Hello, and a very warm welcome to everyone joining us for today's Select Science webinar titled Biomarker Discovery for Lysosomal Storage Disorders Using a Metabolomic Approach. I'm delighted to be joined today by mass spectrometry specialist, Dr. Michelle Boutain. Michelle is the Technical Director of the Waters CHUS Expertise Center in Clinical Mass Spectrometry and Adjunct Professor in the Department of Pediatrics in the Faculty of Medicine and Health Sciences at Sherbrooke University. The Mass Spectrometry Laboratories at the Faculty of Medicine and Health Sciences and Clinical Research Center in Sherbrooke, Quebec, are part of Waters Centers of Innovation. Projects are oriented towards translational research by mass spectrometry biomarker discovery, development and validation of methods and technological transfer to the clinical field. The aim of the Centre is to make a positive difference in the lives of patients and their families. In today's webinar, Michelle will discuss the application of high-accuracy mass spectrometry as a powerful tool for the discovery of disease biomarkers. The presentation will cover the different steps of untargeted and semi-targeted metabolomic studies, as well as strategies to bypass potential pitfalls. Following the presentation, we'll have a question and answer session. So if you have any questions, please feel free to submit these at any time during the webinar, and you can submit these by clicking the purple tab to the left of your screen. You'll also find related materials, such as a downloadable certificate of attendance in the related resources tab to the left of your screen. So without further delay, I'd like to hand over to Michelle and to thank him for presenting to us today. Please go ahead, Michelle.
Thank you for this introduction. This webinar will be separated in two parts. First, I will give general information about untargeted metabolomics, and also I will present some pitfalls that we encountered in our lab during previous studies. During that part, I will talk about sample analysis, so sample collection, sample preparation, UPLC-MS analysis, also data analysis, so data alignment, data scaling, multivariate data analysis, and the structural elucidation of the biomarkers. In the second part of the webinar, I will present two metabolomic studies performed in our lab that were successful to discover new biomarkers. What is untargeted metabolomics? It consists to analyze all the components found in samples, including unknown metabolites, and to compare their abundance between two sample groups. The samples are analyzed directly or with a minimum of separation, such as a liquid-liquid extraction or a solid-phase purification. In general, untargeted metabolomics generates hypotheses, whereas targeted metabolomics tests these hypotheses. Now, our philosophy behind untargeted metabolomic studies to discover biomarkers. In our lab, we prefer to analyze a small number of samples from patients, but patients with severe clinical manifestations, so between 12 and 20 samples from patients and the same number of samples from healthy controls. Why we do that? But first, the small number of samples allow the use of a lower threshold during sample analysis without exceeding the computer capabilities. Also, patients with mild clinical manifestations are often low biomarker excretors and decrease the probability to discover novel biomarkers. For that reason, we prefer to only analyze samples from patients with severe clinical manifestations. After the discovery of good biomarker candidates by untargeted metabolomics, we developed MRM methods to validate these biomarkers and to analyze them in a large cohort of patients when it's possible, over than 200 patients, and this time we include patients with mild clinical manifestations. A word about sample collection. It is important to collect samples from patients and control with the same biological containers at the same site and at the same time, because we don't want to create a bias in the composition of the two sample groups. Molecules from plastic material can be detected and confounded with biomarkers. Unfortunately, it is not always possible to collect controlled samples at the same sites and patient samples. In our lab, we work with very rare genetic diseases. So these samples are very rare. It is even more difficult to obtain samples from patients before the initiation of treatment. So for that reason, often the samples that we use were collected before the initiation of the project. So it was not possible to obtain control samples from the same site. During a previous metabolomic study, we discovered a very promising biomarker. But after a hard work of structural elucidation, we realized that that biomarker was a plasticizer from the tubes used to collect the samples from patients. To decrease the probability of such false positive results, when we collect control samples and other sites than patient samples, we can use different kind of tubes. So tubes with different plastic materials from different companies, so it will decrease the risk that some molecule from the plastic can be found in only one of the two groups analyzed. Concerning sample normalization, in our lab, we normalize urine sample with creatinine or specific gravity. It is important to normalize the concentration of urine sample because the concentration of this sample can vary a lot from one patient to the other or for the same patient during the day. For plasma sample, we normalize using the volume. For cell extracts, we normalize with proteins or cell counts. Without normalization, it is difficult to detect small biomarker variations between the groups. For urine samples, we prefer to adjust the sample volume in function of the normalization before the sample prep to prepare a larger volume of dilute urine samples, instead to apply a correction factor on the results, because if the biomarker is not detected due to the low concentration of the sample, even if we apply a correction factor on the results, the biomarker will remain undetected. Urine sample can be filtrated or not. Some hydrophobic molecules can stick to urinary sediments. For example, when we analyze globotriaosis sphingosine and globotriaosis ceramide as Fabry disease biomarkers, we obtain more sensitive and reliable results by the homogenization of the urine with the sediment instead to filtrate it. Every step of the sample preparation process causing degradation or contamination of a sample group compared to the other may generate false positive biomarkers, and we can waste a lot of time to try to elucidate the structure of these biomarkers. So it is very important to prepare patient and control samples at the same time, and also using the same consumables and solvents to prepare both sample groups. It's also important to prevent the formation of a composition bias between the two sample groups during the sample analysis. So it is important to inject alternatively the two sample groups to prevent biases due to signal decreasing or molecule degradation during the sample run. During the sample run, I also like to inject a QC sample at the beginning, at different intervals, and at the end of the sample list. As QC, I like to take a mixture of all the samples analyzed during the sample batch. So for example, I will take 5 microliters of every samples from patient and every control samples, and I will mix it to generate a pool, and I will inject this pool at different intervals during the sample run. The QC variability must be negligible compared to the biological variability. For untargeted metabolomics, we compare different groups, so the precision is more important than the accuracy. In this first example, we have the concentration of a biomarker in 2 sample groups. In this example, the accuracy was good, So the concentration measure correspond to the reality, but the precision was very bad with very broad distribution. So in that case, it was not possible to distinguish the two sample groups based on that biomarker. In the second example, the accuracy was bad, so the concentration was slightly overestimated, but the precision was very good, and it was possible to differentiate the two groups based on the abundance of that biomarker. So the conclusion of that is that increasing precision allows conclusive results with a smaller number of samples. So it is very important to minimize the variability during sample preparation and sample analysis to increase the probability to obtain conclusive results. It is also important to chromatographically separate structural isomer interferences. For example, in a metabolomic study that we performed in our lab to discover Fabry disease biomarkers, when the samples were analyzed by reverse phase chromatography, G2, galactosyl-ceramide, was not revealed as a Fabry disease biomarker due to its structural interference, lactosyl-ceramide, that was co-eluting with that biomarker. So we have the structure of these two molecules, and the only difference is the conformation of the linkage between the two sugar units. But when exactly the same samples were analyzed in a second metabolomic run using a high-lead column, these two structural isomers were separated, and this time galactosyl-ceramide was revealed as a Fabry disease biomarker. So the conclusion of that is that it is important to perform the metabolomic studies using different chromatographic methods to improve our chances to separate structural isomers and to discover new biomarkers. In a second example, we analyzed glucosyl-ceramide in human brain tissues as a potential Parkinson's disease biomarkers. This time too, we had a problem with a structural isomer interference because glucosyl-ceramide as galactosyl-ceramide as structural interference. The only difference between these two molecules is the conformation of this hydroxyl group. So to check if glucosyl-ceramide is a good Parkinson's disease biomarker, it was very important to separate it from galactosyl-ceramide. I tried the chromatogram showing the separation between these two molecules. It was a big challenge because the matrix, human brain, was very complex, and also the interference galactosyl-ceramide was significantly more abundant than glucosyl-ceramide. Now, data analysis. So I will discuss about data alignment, also called data mining, data scaling, multivariate data analysis, and also the structural elucidation of the biomarkers. Well, data alignment. It consists to classify all the peaks detected by mass spectrometry according to their retention time and their mass to charge ratio. During data alignment, we also evaluate the peak areas and the peak aids. We also remove the isotopes and the metal adducts because we don't want to consider these species as potential biomarkers. If data mining is not performed properly, two different molecules and two different samples may be considered as the same molecule, and the same molecule and two different samples may be considered as two different molecules. So if data mining is not performed properly, it can completely ruin the metabolomic study. For data alignment, we use the marker-link software from Waters. The main parameters to adjust for data alignment are first the intensity threshold. In general, we use very low intensity thresholds like 5 counts or 10 counts because it favors the discovery of very low abundance biomarkers. We also have to adjust a mass windows and a retention time window. So it's tolerances to consider two peaks in two different samples as the same molecule. To set these parameters, I use the QCs analyzed at different intervals during the sample run. In these QCs, I check for some peaks at different retention times and different mass to charge ratios, and I check the minimum and maximum retention time in the different samples for this peak, and the same thing for the mass to charge ratio. And I use these values to set the parameters for data alignment. This is an example of table obtained after data alignment with marker lengths. So we have for each peak detected by mass spectrometry, the retention time, the mass to charge ratio, and the abundance of that peak in the different samples. To improve our chances to discover biomarkers, we can perform data scaling. So with data scaling, we transform the peak area of the peak height of the markers according to a mathematical algorithm. At the example in left, we have an example of chromatogram without scaling. So without scaling, the most abundant markers will be statistically favored. But if we use the Pareto algorithm, with the Pareto algorithm, we mathematically modify the peak area measured for the marker. So the new peak area becomes the peak area measured for that marker, minus the mean peak area of that marker in the different samples, divided by the square root of the standard deviation of the peak area of this marker in the different samples. With the Pareto algorithm, we will artificially decrease the abundance of the most abundant molecules without significantly increase the abundance of the low abundance molecules. So with the Pareto algorithm, the molecules with a medium abundance will be statistically favored. Using the UV or unit variance algorithm, this algorithm is very similar to Pareto, but without the square root on the standard deviation. With the UV algorithm, we mathematically decrease the abundance of the most abundant molecules, also the abundance of the medium abundance molecules. So this algorithm will favor the metabolites of low abundance, but it will also significantly increase the background noise. All these data scalings are performed with the EasyInfo software from Umetrix. This software is integrated to the toolbox of marker links. During an untargeted metabolomic study, we don't know if the biomarkers are of low, medium, or high abundance. For that reason, it is interesting to check all the algorithms, to try all the algorithms to improve our chances to discover biomarkers. The multivariate data analysis was also done using the EasyInfo software. I'd like to start with the principal component analysis to test the reproducibility of the analysis. PCA is an unsupervised data analysis giving an equal weight to all the peaks detected in the samples. So on the score plot, each point corresponds to a different injection on the mass spec. So the black points to the injection of patient samples and control samples. And in blue, we have the replicates injection of the QCs. So more two points are close together on the score plot, more their composition is similar. So since the QCs are replicate analysis of the same sample, we expect to detect QCs, the point corresponding to the QCs, very close together. If it's not the case, we had a problem of reproducibility during the analysis, or the data alignment was not performed properly. Also, the QC is a mixture of all the samples analyzed during the sample run. So for that reason, we expect to detect the points corresponding to the QC in the middle of the cloud of points corresponding to the samples. Thereafter, we can perform an orthogonal partial least square discriminant analysis. This time, this is a supervised data analysis giving more weight to the peak differential expressed in the two sample groups. On the score plot, each point corresponds to a different sample. The green ones the control, the red ones the patient. As you can see, both groups are completely discriminated. It means that at least some molecules are good to differentiate the two groups, and these molecules are the potential biomarkers. To find these biomarkers, we can generate a F-plot. On the F-plot, each point corresponds to a marker aligned using marker links. More the marker is at the top right of the F-plot, more this molecule is abundant in samples from patients and absent in samples from controls. At the reverse, at the bottom left, the more the point is at the bottom left, more the marker is abundant in samples from controls and absent in samples from patients. So the potential biomarkers are at both extremities of the F-plot. Thereafter, it's important to check if the points at the extremities are good biomarker candidates. To do that, we just have to click on the point and it generates a trend plot. On the trend plot, on the y-axis, we have the abundance of the biomarker, and on the x-axis, the different samples. So the black points correspond to the controls, the red points to the patients. In this example, we have a good biomarker candidate because the biomarker was present in all the samples from patients and absent in all the samples from controls. In this second example, the biomarker is more abundant in samples from patients, but it is not present in the samples of all the patients, and it is also present in two control samples. So that biomarker was rejected. It is also important to verify that the biomarker is not an interference from the mobile phases or from the UPLC system using the extracted ion chromatogram. So in this first example, when we extracted the biomarker from a sample, we obtain a peak. It's what we want to see. But in this second example, the biomarker was not really a peak, but a fluctuation in the signal of an interference. So it's not really a biomarker, it's a false positive result. And such kind of the probability of such kind of false positive result decrease if we increase the number of samples during the metabolomic study. For the structural elucidation of the biomarkers, we perform tandem mass spectrometry experiments using our Synapse G1Q-Tof. So we fragment the biomarker, we isolate the biomarker in the quadrupole, we fragment it in the trap, and we analyze the fragments using the Tof detector. So we have accurate mass measurement for the fragments, so we can obtain the empirical formula of the fragments, and it is very useful to elucidate their structure. To help me with the structural elucidation of unknown molecules, I also like to use molecular databases such as ChemSpider. This database is free. It contains more than 89 million structures. It regroups 274 data sources. Molecular databases give a list of possible structures for an unknown molecule, but we must keep in mind that the biomarkers detected during a metabolomic study may not be reported in databases. Also, some databases contain synthetic molecules in addition to biological molecules, so we must take care of that. For example, if we detected a biomarker with a mass-to-charge ratio of 219, with an accurate mass measurement and thus the empirical formula, with ChemSpider, there is 1,048 possible structures for that molecule. Without accurate mass measurement, so only with a unit mass measurement, we have more than 35,000 possible structures. So it is very useful to have accurate mass measurements when we want to elucidate the structure of an unknown molecule. I know that even with accurate mass measurement, 1048 possible structures is very high. But we must keep in mind that each time that we elucidate a part of the structure of the molecule by tandem mass spectrometry, or each time that we find a chemical function, we elucidate a chemical function in the structure of the molecule, each time it decreases the possibilities of structures, and it can decrease very rapidly. I think that it is also important to mention that as for drugs, endogenous molecules can be metabolized in the liver before the excretion in urine. That metabolization occurs in two phases. In phase 1, we have reactions to increase the polarity of the molecule, like oxidation, hydrolysis, cyclization. It increases the polarity of the molecule and thus favor its excretion in urine. In phase 2 metabolization, we have a reaction of conjugation and detoxification, such as the addition of glucuronic acid, sulfonate, or glutathione. Each time that we discover a biomarker by untargeted metabolomics, it is interesting to check if this biomarker has also metabolized excreted in urine, or if this biomarker is a metabolite of another molecule. So from the discovery of one biomarker, sometimes we can discover a family of biomarkers. To help us with that, we can use the Metabolomic software from Waters. This software is specialized in the detection and quantification of drug metabolites and might be useful to discover metabolites or analogs of a biomarker found during a metabolomic study. The software will calculate the mass of the possible metabolites for the molecule that we want to test, and will also check if these metabolites are present in the samples. For example, we have here the structure of Lyso-Gb3, a biomarker of Fabry disease, and we found 4 of its metabolites in urine, and these metabolites were more abundant in urine than Lyso-Gb3 itself. Now I will start the second part of this webinar. So I will present 2 metabolomic studies performed in our lab to discover Fabry disease biomarkers in urine. So during this part, I will briefly introduce Fabry disease. I will present a metabolomic study targeting basic molecules, another metabolomic study targeting hydrophobic molecules, and I will give a general conclusion. Fabry disease is an X-linked lysosomal storage disorder. So in general, the disease is more severe for males compared to females. It is caused by an alpha-galactosidase A deficiency. The function of this enzyme is to cleave the large sugar unit of globotriaosylceramide, a sphingolipid found in membrane cells, in membranes from cells. So the enzyme deficiency causes storage of glycosphingolipids such as Gb3 and related molecules. Now the main clinical manifestation of Fabry disease. For children, there is acroparesthesia, so intense pain at the levels of hands and feet. Hyperhidrosis, so decreased sweating. Heat and cold intolerance. Corneal opacities. For adults, there is early ischemic stroke, ventricular hypertrophy, progressive renal insufficiency conducting to dialysis, and also angiokeratoma, so some red lesions on the skin. Before our metabolomic study, the Fabry disease biomarkers that were mostly used for the diagnosis and high-risk screening of the disease in urine and plasma were Gb3, so the substrate of the deficient enzyme itself, and also Lyso-Gb3. This is the same molecule but without the fatty acid moiety. During a first metabolomic study, we targeted basic molecules, so it includes Lyso-Gb3, so that non-biomarker was like a positive control in this metabolomic study. So for this study, I analyzed urine samples from 18 untreated Fabry males, 45 untreated Fabry females, 22 LT controlled males, and 36 LT controlled females. For the discovery of the biomarkers, only samples from males were used because, as I mentioned before, the disease is more severe for males compared to females. So we expected higher levels of biomarkers in urine samples from males compared to females. The samples from females were used only for the validation of the biomarkers. For the sample preparation, I did a solid phase extraction with a mixed mode cation exchange cartridge. It was OSS cartridges from Waters. For the sample analysis, I use our Synapse Q-Tof. For the liquid chromatography, I use a C18 column. And for the mass spectrometry analysis, I use the Tof detector. I use the log mass option, so the instrument was recalibrated at every 5 seconds during the sample run, and the calibrant was sulfinadim. This is an example of chromatogram obtained during that metabolomic study. After data alignment using the marker link software, we had more than 50,000 markers. We use a very low threshold for data analysis, a threshold of 5. I tried different thresholds and I had to decrease the threshold up to 5 to detect our positive control, the already known biomarker Lyso-Gb3. For this study, I obtained better results using the univariate algorithm because the biomarker were at low abundance. This is the S-plot obtained during that metabolomic study. So we detected eight potential biomarkers. Among these biomarkers, we have Lyso-Gb3, our positive control. To validate the identification of Lyso-Gb3, we did its tandem mass spectrometry analysis. So this is the spectrum. So we have the molecular ion, the loss of 1, 2, and 3 sugar units, and also the peak corresponding to the dehydrated sphingosine. When we analyzed another biomarker, we were surprised to observe a similar fragmentation pattern. So this molecule was a Lyso-Gb3 analog, but with a modification on the sphingosine moiety. So that molecule was Lyso-Gb3, but with only 16 carbon on the sphingosine moiety instead of 18. In fact, all the biomarkers detected during that metabolomic study were Lyso-Gb3 analogs with modification on the sphingosine moiety. So we detected modification corresponding to -12, -2, +14, +16, +34, and +50. I took the accurate mass measurement of all these modified, with all these biomarkers with modification on the sphingosine, and I subtracted from that accurate mass the accurate mass of Lyso-Gb3. So I obtained with that the accurate mass of only the modification on the sphingosine, and with that it was possible to obtain the atomic composition of these modifications. So we don't know exactly what are these molecules, but at least we know that this is Lyso-Gb3 analogs with modification on the sphingosine chain, and we have the atoms involved in these modifications. Also, the retention time for these Lyso-Gb3 analogs correlate with the polarity of the modification on the sphingosine chain. Thereafter, we analyzed these biomarkers in all our Fabry samples, so including samples from Fabry females. Something interesting to mention is that the new biomarkers are more abundant than Lyso-Gb3 itself. In the case of the biomarker with a mass to charge ratio of 802, its abundance was 17 times the abundance of Lyso-Gb3 in urine. So this new biomarker is significantly more sensitive for the diagnosis of the disease than Lyso-Gb3, the biomarker previously used. In conclusion, this metabolomic study revealed 7 novel Fabry disease biomarkers corresponding to Lyso-Gb3 with modified sphingosine moieties. We found the empirical formula of these modifications on the sphingosine. To show the clinical utility of these new biomarkers, something that is important to mention is that for Fabry disease, there is some mutations called late onset cardiac variant mutations. Patients with these mutations have less clinical manifestation than patients with classical Fabry mutations, but they have some cardiac involvement. And patients with these mutations are very low biomarker excretors. So using only Lyso-Gb3 to diagnose these patients, often many patients were missed. But with the new biomarkers, especially the one with a mass to charge ratio of 802, we can diagnose more patients with the cardiac variant mutation than Lyso-Gb3 itself. Also, it was possible to correlate the left ventricular hypertrophy of these patients with the levels of some Lyso-Gb3 analogs in urine, and it was also possible to correlate these biomarkers with the main severity score index. It's an index to evaluate the clinical manifestations of Fabry disease. Now, the second metabolomic study, this time we targeted hydrophobic molecule, and Gb3 was used as positive control for this metabolomic study. The previous metabolomic study shows that we can detect chemical modification on the sphingosine moiety of Lyso-Gb3. So we can expect similar modifications on the sphingosine moiety of Gb3, but this time these modifications will be declined in many molecules due to various fatty acid moieties. For this study, the samples were purified by liquid-liquid extraction with chloroform, and the organic phase was analyzed. This time we obtained better results with the Pareto algorithm because the biomarkers were of medium abundance. This is the S-plot obtained during this study. So it revealed 18 Fabry disease biomarkers. After structural elucidation, we realized that all these biomarkers were structurally related to Gb3. These biomarkers can be subdivided in 5 groups. In the first group, we have Gb3 molecule with different fatty acid chains. This was the positive control of this study because these biomarkers were already known. So to show the structural elucidation, first we have the molecular ion, we have the loss of 1 water molecule, the loss of 1 sugar unit, 2 sugar units, 3 sugar units to generate the ceramide. We have the peak corresponding to the fatty acid moiety and the peak corresponding to the sphingosine moiety. For the other groups of biomarkers, I will only present the peaks corresponding to the fatty acid and to the sphingosine because the other peaks were the same for all the molecules. Okay, now the second group of biomarkers, it corresponds to Gb3 molecules with an extradable bond. These biomarkers were difficult to structurally elucidate because, in fact, it was not only one molecule, but two co-eluting structural isomers that were fragmented together. So in red, we have the molecule with the extradable bond on the sphingosine moiety. And this is the peaks corresponding to this structural isomer. In blue, we have the molecule with the extradable bond on the fatty acid moiety. And this is the peaks corresponding to the fragment of this molecule. Now, the third group of biomarkers, this group corresponds to Gb3 molecules with two extradable bonds. So in red, we have the molecule with one extradable bond on the sphingosine and one extradable bond on the fatty acid. And in blue, we have both extradable bonds on the fatty acid moiety. It's important to mention that it was the first time in group 2 and group 3 that we detected an extradable bond on the sphingosine. This is a modification that was also observed on Lyso-Gb3 in the previous study, but it was the first report of that biomarker for Gb3. Now the group 4, it corresponds to Gb3 molecules with an hydrated double bond on the sphingosine. So this is another modification that was also observed on Lyso-Gb3, but this is the first report of that modification on Gb3. Now the last group of biomarkers, this was the big surprise in this study because these biomarkers were not expected at all. So we discovered Gb3 molecules with a methylation of the amide linkage. It was possible to locate the methyl group at that position because it was detected on the fragment corresponding to the fatty acid, but also on the fragment corresponding to the sphingosine moiety. And the only common atom between these two fragments is the nitrogen atom, so the methyl group must be located there. In conclusion, we detected 18 Gb3-related Fabry disease biomarkers in urine. Among these biomarkers, first, some were already known, but we detected some Gb3 molecules with modification on the sphingosine as supplementary bonds, double bonds, and hydration that were not reported in literature before. Also, we discovered 7 methylated Gb3 isoforms. These results highlight the importance to evaluate a profile of biomarkers because these different groups, the relative abundance of these groups of biomarkers vary from one patient to the other and also within the time for the same patient. So it is very important to check all this profile of biomarkers to have a complete overview of the health state of the patient and not to see only a piece of the pie. We have an hypothesis concerning methylated Gb3. We think that it can be an intermediate conducting to the deacylation of Gb3 to generate Lyso-Gb3 because the biological origin of Lyso-Gb3 is not well known. So we want to perform a test in future to validate this hypothesis. To conclude, I'd like to thank Cristiano Reble, the director of the lab. He is also the scientific director of the Water Center of Innovation. I'd like to thank my colleagues, the graduate students involved in this project. For financial support, I would like to thank Waters Corporation, La Fondation du CHU, CIHR, Sanofi Genzyme. I also would like to thank you for your attention, and I will be happy to answer to your questions. Thank you.
Thank you very much, Michelle, for a very interesting presentation. It's now time for our question and answer session. So to our audience, if you have any questions, please submit them now using the purple button to the left of your screen. If you click on that tab, you'll be able to type in your question. So the first question we have here, please can you comment on the use of urinary Gb3, lyso-Gb3 and their metabolites as markers of Fabry disease versus enzyme activity assays that are performed from newborn dried blood spots?
Thank you for this question. The analysis of enzyme activity is very good for the diagnosis of classic Fabry males, but it is not very good for patients with residual enzyme activity, such as female and males with some specific mutations. So I really think that some of the new biomarkers, especially the lyso-Gb3 analogs in urine, are more sensitive for the diagnosis of the disease for patients with residual enzyme activity than the analysis of enzyme activity in dry blood spots.
Great, thank you. The next question we have here, is there a different non-polar marker for the normalization of urine samples? I understand that creatinine may behave differently because it's very polar and most lysosomal biomarkers are very non-polar.
It's a very interesting point. In our lab, we always use creatinine for the normalization of the urine samples. I know that in some labs they use the specific gravity. I also read some publications where some non-polar molecules such as ceramide or sphingomyelins were used for the normalization, but the problem is that these molecules are very closely related and in the some biological pathways than the molecules that we want to analyze. So it's the reason why we didn't use these molecules to normalize urine samples. But it's a good point to study for the normalization of urine samples.
Okay, thank you, Michelle. The next question. From your relative quantification plots, it appears that some Fabry males and Fabry females have lyso-Gb3 concentrations that are not different from those of control males and females. Can you comment on the impact this has had on the sensitivity and specificity of the assay?
Yes. In the results that were presented concerning lyso-Gb3 analogs in urine, the biomarkers were not detected at all in the control samples. But some biomarkers were not detected in the patient samples also. But it's important to mention that it was only the result for the discovery phase of the project. Thereafter, we developed a more sensitive method by MRM using our triple quad. This method is more sensitive, so the difference between the control and the Fabry patient is more statistically different. With these biomarkers, the sensitivity is very good. Only some patients with very, very low clinical manifestation and very high residual enzyme activity are not detected by these biomarkers.
Great, thank you. We've got time for some more questions, so if there are any more questions from our audience, please feel free to submit those now by clicking on the purple tab. The next question here. Peak picking programs, such as Progenesis, often pick up noise when the threshold is lowered. What strategies are there to more quickly discard noise so that one can focus on real peaks?
I know that the software took a look at peaks corresponding to noise, but the statistical analysis, the S-plot, removed the vast majority of these false positive results. So I really think it's better to use a low threshold, even if we have the peak picking of some background noise, because the noise is removed during the statistical analysis. sometimes we have to remove one or two false positive results manually, but I really think that it improves our chances to discover interesting biomarker by using a very low threshold.
Great, thank you. The next question here, to achieve mass accuracy of under 10 parts per million, how often is the mass calibration needed?
For the metabolomic study, we have a calibration that is working during the sample analysis. So the instrument is calibrated every 5 seconds during the sample run using the log mass, because we have a standard terphenadine with a known mass that is infused at the same time of the samples, and that molecule is used for the calibration at every 5 seconds.
Great, thank you. The next question here, what are the characteristics of GB3-ME and GB3 deacylated?
Would you please repeat the question?
Yes. What are the characteristics of GB3, I think methylated, and GB3 deacetylated?
The GB3 is less polar than lyso-GB3. The deacylated form liberates an amine function, so it modifies the chemical properties. So lyso-GB3 is basic, it contains an amine function, so it's why it was a good way to purify it, to use a M6 cartridge from water, so a strong cation exchange cartridge. And GB3, with GB3 there is a fatty acid connected to that nitrogen atom, so we lost the basic property of the molecule.
Great, thank you, Michelle. The next question here, what is the next step following your initial biomarker validation work? Have any been implemented for screening in your lab so far?
After the discovery of the biomarker, we developed high sensitivity methods using our triple quad, so MRM methods. And some of these biomarkers are already used in different projects. For example, we analyze twice a year urine and plasma samples from Fabry patients enrolled in the Canadian Fabry Disease Initiative project. So it consists of 550 Fabry patients, and we analyze lyso-GB3 analogs in urine and plasma for these patients twice a year to monitor their health state, and also to monitor their response to treatment. Also, we use the new biomarkers, especially the lyso-GB3 analogs in urine, for high-risk screening. So we receive urine samples from patients presenting clinical manifestations that can be associated to Fabry disease, such as corneal opacity, ventricular hypertrophy, and renal insufficiency. And we use the biomarkers to check if the reasons for these clinical manifestations are associated to Fabry disease. And using that approach, we diagnose some Fabry disease patients, and the disease was confirmed thereafter for these patients using mutation analysis.
Thank you. And the next question here, was there a reason that SPE was used for sample prep? Is there concern that SPE might remove biomarkers?
The SPE was very effective to collect all the molecules with an amine function, and so it was very appropriate to analyze lyso-GB3.
Great, thank you. And the next question here, how many novel biomarkers have you discovered related to LSD disorders using this method so far?
For Fabry disease, we performed 5 different metabolomic studies, 3 in urine, 2 in plasma. We detected a total of 71 different biomarkers. More than half of these biomarkers were not reported in literature before, so were discovered in our lab. Also, we work on Gaucher disease. We have a publication that is in press and will be available soon concerning the discovery of Gaucher disease biomarkers.
Great, thank you. And the next question here, how did you validate the biomarker quality?
But after the discovery of the biomarker, we developed a very sensitive MRM method. These methods are quantitative with a calibration curve, and we use that method to analyze more than 100 samples from Fabry patients, so patients with mild clinical manifestations, treated patients, and we compare statistically the results obtained for the Fabry patients and the healthy controls, and the sensitivity of the method for the diagnosis is very high.
Great, thank you. That's all we have time for today. So thank you very much to Michelle for today's informative webinar, and thank you to everyone joining us online for sending in your interesting questions. If we didn't have time to answer your questions, or if you have a question that you'd like to ask, please feel free to submit that now and we'll follow up with them after today's event. You can submit the questions by clicking on that purple tab to the left of your screen, or you can email me at editor@selectscience.net and I will follow up with your questions. Remember, you can also download a certificate of attendance in the related resources tab. And if you'd like to listen to today's webinar again or to invite a colleague to listen, it will be available to watch on demand in a few days' time. So goodbye and thank you once again for everyone joining us.